right, next we have genetics, actually specifically molecular genetics and how your DNA determines your traits. Remember that the making of proteins is what this is all about. You inherited an order of bases in your DNA that was specific to you, unique to you. That DNA is used to make mRNA in the nucleus. And that mRNA leaves the nucleus and goes to the ribosomes to make a protein. So we have first our DNA, that's used to make mRNA, and that is used to make a protein. Let's look back at the structures and what this looks like. So we inherit DNA, series of T's, G's, C's, and A's. You can tell that this blue molecule, number 1, is DNA because it has A, T, C, and G. The red one, you can tell is RNA because it has U in it. It has A, U, G, and C. In DNA, A matches to T and C matches to G. RNA, U takes the place of T. Okay, so we have our DNA, and let's say this person inherited AAACGT. That order of bases would then be used to make an order of bases on mRNA that match. A matches to U, C matches to G, T matches to A. So you would get UUU, GCA. That process that happens in the nucleus where the DNA is, and DNA never ever leaves the nucleus, that process is called transcription. We're transcribing the order of bases in DNA into an order of bases in mRNA. The mRNA then leaves. It takes the message. It's a messenger RNA and takes it to where the proteins are made in the ribosomes. So now we have that same exact mRNA that was made by transcription sitting in the ribosome, in the location of the ribosome, and that mRNA is being used to make a protein. The protein is made when these tRNAs, these green things, carrying the amino acid come in and match. If this is UUU, that is AAA, and it's carrying a specific amino acid called phenylalanine. You don't need to know what the name of the amino acid is. We use a chart for that. But the order of bases in the DNA that you inherited that determine the order of bases of the mRNA is what's going to tell you what amino acids to put together and therefore what protein to make, because remember, proteins are made of amino acids. And the proteins basically make you look the way you do. They're either enzymes that control reactions or they're colorful proteins in your eyes or your hair or your skin, and they give you all your traits. So to recap, DNA makes the mRNA by transcription in the nucleus, and then translation happens at the ribosome when the mRNA is used to determine the order of tRNAs, which carry the protein parts called amino acids to make a protein that determines what you look like. Scientists have taken advantage of that by doing genetic engineering. Now that we know that DNA determines the proteins that are made, we can just take out the DNA from an organism that makes a protein we want and cut it out and paste it into another cell and have that cell make the protein. We use enzymes to do the cutting and pasting. So we could take a human gene for insulin and put it into a bacterial cell with bacterial DNA and make the bacterium make insulin. And it will just produce insulin. Now, it doesn't have a bloodstream and sugar to control or anything. The insulin's useless to the bacterium, but it'll produce it and we can kill the bacterium and take the insulin and give it to diabetics. There are a lot of different ways that we can cut DNA from one species, put it in another, and make that new species make the protein from the donor. Another bit of knowledge that we've got from genetics is about cloning. We have been able to clone animals. We can take the whole nucleus from a sheep, like let's say we wanted a little black sheep. We could take the nucleus out of one of its cells and put it in an egg cell in a zygote. Take the nucleus out of the zygote and put the nucleus in from the organism we want to copy. And that zygote now has all the information from that cell that got that nucleus, and it will grow into a baby that has all those traits that were in that nucleus that came from that donor. We can implant it in a mama in her uterus, and she can carry the baby around. And when that baby is born, it's going to have all the same DNA that was in the nucleus that we took from the original donor. We've been able to clone this sheep, something that asexual reproduction normally wouldn't happen for a sheep. Keep in mind, genetic engineering increases variation. We create new varieties. Cloning decreases variation. We are basically simulating asexual reproduction with no variation. And remember, sexual reproduction, variation, asexual reproduction, no variation.